Okay, we're gonna get started with Ana Maria's keynote. Thank you all for joining us this morning. I want to introduce her. I'm so excited that she accepted our invitation to come here. I've been following / stalking her on Twitter for a while. Dr. Ana Maria Porras is an assistant professor of biomedical engineering at the University of Florida, where she leads the Tissue Microbe Interactions Lab. Excuse me. Her group engineers models of disease to study human microbe interactions in the context of the gut microbiome, global health, and neglected tropical diseases. She's also a science artist and an expert on inclusive bilingual science communication and in BASTA. Dr. Porras is the co-founder and senior advisor of the Latinx in biomedical engineering community. She was selected as an if-then AAS AAAS ambassador for girls and women in STEM and was honored with a statue at the Smithsonian Institution within that program. Prior to arriving in Florida, Dr. Porras was a presidential postdoctoral fellow at Cornell University. She holds a bachelor's in biomedical engineering from the University of Texas at Austin and a master and a PhD from the University of Wisconsin-Madison, where she was also an American Heart Association predoctoral fellow. When she isn't doing all that, she loves to read, dance, travel, and above all, eat ice cream. So, welcome back to Wisconsin.
Gracias, Claudia. Buenos días. Hi, everyone. Good morning. Thank you for joining us this early in the morning, and thank you for being here. Before we start, I like to say this general disclaimer. I suffer from an essential tremor. So when I'm a little nervous, my hands get a little bit out of control sometimes. I'm a little nervous today, and I can feel my hands are a little bit out of control. So if you see me shaking, I'm okay. It's just my body's natural reaction to adrenaline and nerves. Okay, and I want to start by thanking you, Claudia, for inviting me. This is, my sister took this picture when I graduated. This was I guess I walked in 2017, but I actually defended my PhD at the end of 2016. And I was here in Madison for 6 years. So it's kind of cool to be doing it in this building too. When I started my PhD in 2011, the WIT wasn't open yet. It opened at the end of that year. Union South had just opened. There was no food. So it's really cool to be back. And also as an international student, I always wondered why you would come back on homecoming. And so it is extra cool that I'm here giving this keynote during homecoming weekend. And so Claudia asked me to talk about both the research that I do and kind of my trajectory and how I got there. And I think at least one of you was at SACNAS last year. Was anybody else at SACNAS? No. Okay, a couple of people. Some of you already saw some of these on my keynote at SACNAS last year, but I thought it was really appropriate to adapt that shorter keynote to this, because really the story of how I got to where I got is really incomplete if I don't tell the story of the Colombian Badgers. And it's just really fitting that this is our theme today, or the Latinx community in STEM here, right here at the University of Wisconsin, because it was that community that actually changed the trajectory of what I study and of the way that I do science. And I'll explain that more in a second. Also, forgive the bad resolution. It was like 2015. Our phones didn't really have such good cameras anymore. And I'm staying with Gina after this, the rest of this weekend, and I'm seeing a lot of the people in this picture throughout this weekend. So I was born and raised in Bucaramanga. Raise your hand if you've ever heard of it. Yeah, like a few people, awesome. So Bucaramanga is quote unquote small. It's no longer that small, it's like top 5 in Colombia. It's a city of about 1 million people. That's small though, as many of you know, for Latin American standards. Pretty big for American standards, for city sizes, in the middle of the Andes. My sister and I grew up, it's just the two of us, grew up in kind of an unusual household in that my parents, Hernan Porras and Marta Corredor, pictured here, they were both first-generation college students who later managed to become engineering professors. So we just grew up in a different environment. I grew up like, I didn't know, I still don't think being a professor is a particularly exceptional job. It's just a job that gets glorified by society. But like my parents and all their friends were professors. So like to me, it was just like, and not to mention in Spanish, there is no difference between being a high school teacher and a university teacher. It's just profesor, right? So I feel like in English, there's this additional prestige that goes with the word that kind of goes away in Spanish. Also, I noticed this yesterday. Sorry, super random aside. Like, do you notice my shirt? I have like a Minnie Mouse shirt. I think I was destined to live in Florida. I like gifted my friend a Disney annual pass for submitting the NSF career this summer. So now I'm like a Disney adult. So, and anyways, if you're in Disney in the coming year, let me know. I'll go with you wherever. I have an annual pass. But based on the introduction and on what I just told you about my parents, you are probably thinking that I always knew I wanted to be an engineer, and that actually could not be further from the truth. I graduated high school, and I had no idea what I wanted to study. Literally graduated, went on a year abroad as an exchange student in Germany because I didn't know what I wanted to study. And in Colombia, it's not like the US where you can just, and I think it's the same in all of Latin America, where you can't just go to school and be like, oh, I'm undeclared. You like have to know what you want to study. And it was actually 3 months after I graduated high school. My mom traveled to another university, heard that biological engineering existed, and that was the first time both her and I had ever heard that you could do biology and engineering together. And that already kind of tells you a lot about the state of biomedical engineering in the entire country of Colombia in 2005, because even 2 engineering faculty did not know that biomedical engineering as a discipline existed. And so there were only 2 programs in the whole country. That has changed a lot since. But it's for that reason that I actually moved to the United States. So we made kind of the collective decision. At the time, the dollar was not as expensive as it is now, so the conversion wasn't as dramatic. And I ended up moving to the United States for college. I went to, as was mentioned earlier, I went to college at the University of Texas at Austin. I think this is maybe my second year. So baby me. And the first year was hard. So yes, my parents were professors. They could help me with some things. But as I think a lot of you know, because you are not from here, there's so many things about the American higher education system that are so uniquely American that like nobody can prepare you for. So for example, I didn't really know it was important to join student organizations until I was a junior. That's when I joined my first one. And then of course, how many of you are or have been in the past international students? Yeah, so like a good chunk of you. There's like a ton of other stuff and regulations and things that you're just like not aware of. I actually don't remember anything from the first week that I was at UT, except getting lost. But it was bad that I didn't remember because I, for example, completely erased from my brain that you needed to have your I-20 re-signed yearly. So the first time I re-entered the country, I did not have an updated signature because I forgot. The officer was really nice in Houston, but anyways, an example of like how that first week was a little bit, like it wasn't bad, I just don't remember. I think it was just too much. But slowly and surely, I like gained my footing in college. In my second year, I feel like a lot of things really changed for me. I started tutoring for the learning center. That's where I discovered that I love to teach. I still love to teach. It is the reason why I am a professor. It is my favorite part of the job, way more so than the research, even though I also love the research. And I also, thanks to a Women in Engineering program, joined a research lab through the Graduates Linked with Undergraduates research program. And I realized this morning that I forgot to add her picture here. I joined Dr. Christine Schmidt's lab at the University of Texas at Austin. And while I'm not gonna go super deep into the research I did there, it's important because Christine later moved to Florida, and up until 3 months ago, she was my chair at the University of Florida. So and I say that a lot, I bring that up all the time for the students in the room, because this is also why it's important to stay connected and to like put your best foot forward always, because you never know, you literally never know how like life will come back around and you'll like either will meet people again or who is gonna give you an opportunity later in your life. So in Christine's lab, I learned that I was really interested in 2 things. One, this idea of tissue engineering. So tissue engineering is kind of the biomedical engineering solution, and I want to say solution in quotes because the promise of tissue engineering has not really materialized because it's really hard to do. Some people in this building work on tissue engineering. A lot of people on campus work on tissue engineering on this campus. But the traditional premise of tissue engineering is this idea that you would get some sort of cell source, maybe from the patient themselves, maybe from a donor. You would put all of that inside of some sort of material, maybe a biomaterial that's inspired by what's already in your body, that kind of provide the structure of what an actual tissue would look like. You would add a bunch of biological factors to allow those cells to grow, and you could grow like a mini tissue in the lab, and then you could put it in a patient, and that way we wouldn't, for example, need organ transplants anymore. That was kind of the original premise of the field. This has worked with some success on some tissues. So for example, if you have a really bad skin burn, you do find commercial tissue engineering tissue engineered skin that you can use rather than having like a donor skin graft that's used in the clinic all the time. For a lot of organs, we now know it's actually really hard to do, but that's the general premise of tissue engineering. And I had one of my cousins who has since passed away, was much older than me. When she was a teenager, she had a kidney transplant. So I was just fascinated by this idea of like, what if people didn't need, like what if my cousin didn't need a second kidney transplant, et cetera. So that's what attracted me to this field. And it's a field I still work in now with a slightly different application. The other thing I became kind of obsessed with, and this has been a theme throughout my entire career, is the extracellular matrix. And that's kind of a really long, fancy word to say everything, right? So we think of tissues, and most of the time we think a tissue is composed of cells. And that's not true. Most of the tissue is actually composed of extracellular matrix, which is all the stuff that is outside of the cell. So think of it as like a chocolate chip cookie, right? The chips are the cells, the dough, that's your extracellular matrix. And if you think about it, right, you could have a chocolate chip cookie, but you could also have a chocolate chip bread. What makes the cookie different from the bread? It's the dough, the quality of the dough, the properties of the dough, how hard the dough is, how it feels when you bite it. It's similar for the extracellular matrix and human tissues. It's the properties of the extracellular matrix that kind of gives tissues their mechanical properties, and also because cells can have receptors for the molecules in the extracellular matrix. They can communicate with that extracellular matrix, and that matrix can also guide cell behavior. So it has a lot of consequences. Most diseases in the human body, there are some sort of changes in the extracellular matrix. You just don't hear a ton about them. And you also, a lot of the times people are like, oh, I've never heard of that before. I'm like, I guarantee you that you have. Obviously, maybe not in the words of extracellular matrix, but most of us have heard of the components of the extracellular matrix. How many of you have heard of collagen before? Right? How many of you have heard of hyaluronic acid? The women will raise their hands because it's a very common product on skincare, right? Like just this morning, I put some hyaluronic acid serum on my face, right? So we've heard of these molecules a lot. They're like out there, they're in cosmetics a lot, and they play a really important role in our biology. I'm gonna give you 2 examples later where you'll see why I find them so fascinating. And so I learned that I love to do research. I learned that I love to teach in college. And so I knew I wanted to be a professor. And the main reason I did a PhD is because in the United States, and honestly increasingly more so in Colombia, you kind of need one if you want to teach at the university level. So yeah, I didn't really do it because I love to do research, which is what people always write that in their personal statements. And in my head, I'm like, but why do you really want to do a PhD? And in my case, it's because I knew I needed one, like quite explicitly. So, but I got really lucky and landed here at Wisconsin. Wisconsin wasn't even on my radar when I was applying. But I ended up here with Kristen Masters, who sadly for Wisconsin left UW earlier this summer to become the chair of the BME department at the University of Colorado, Denver. I see some people nodding because if you know Kristen, you know how big of a loss that was for the Wisconsin community. And Kristen is an incredibly wonderful mentor. And I honestly joined her lab primarily because, yes, she was doing tissue engineering work, but I always say that that is the only time in my life that I have experienced love at first sight. and it was mutual. Like at our first meeting, I was just like, oh my gosh, you're amazing. And I kept going on recruiting visits, and I would meet people, I'd be like, they're awesome, but they're not Kristen. So I ended up in her lab, and it ended up being just as awesome as I thought it would be. And the thing that Kristen taught me is that we can use tissue engineering, but not what about if we don't use it to put stuff in a patient's body? But what about if we use it to something similar to what you said yesterday, Daniel, right? You talked a lot about models and theoretical models and how you like simplify the world into these theoretical models. So we use tissue engineering to create simplified models of disease, physical models of disease, so models of disease tissues, but in the lab. So that way we can kind of bridge the gap between cells on plastic, and plastic has nothing to do with our bodies, and animals, right? So we can kind of bridge that. And Kristen does that in the context of the aortic valve. Okay, so the aortic valve is a really fascinating organ. It's incredibly thin. It consists of 3 leaflets, 3 triangles that open and close billions of times in your lifetime to control the flow of blood from your heart to the rest of your body, okay? Literally will open and close billions of times in your lifetime. It is open and closing as we speak. It's keeping you alive right now. And another thing that's super fascinating about heart valves is that even though it is very, very thin, so like literally thinner than like a tissue, okay? Fun fact, I used to drive to New Glarus every other Thursday to go get pig hearts for our research. Every other Thursday. It was a fun time. So one thing that is always has amazed me about heart valves is that even though it is such a thin organ, it is incredibly stratified. So if we look at the extracellular, this is staining of the extracellular matrix of a cross-section of a leaflet. You can see in purple, these are elastin fibers. So it's one of the components of the extracellular matrix, and as its name implies, they stretch, and it's what allows the valve to fully open and to fully close and fully open. Then in the center, we have glycosaminoglycans. An example of a glycosaminoglycan is hyaluronic acid, okay? Do people know why do we have hyaluronic acid on face creams, for example? What's its purpose? Does anyone know? You need to watch more makeup TikToks. Yes, okay, some people do watch makeup TikToks, yes. It's for hydration and for water. It is very hydrophilic, it attracts a lot of water, it is very hydrating, and water is really important because it provides a compressive cushion. Because if you think about it, the water is flowing through the valve at really fast speeds, you need a compressive cushion for the cells. And it's always, going forward, it's always gonna be stained blue. And then at the top, you see collagen. And so collagen is another one of these proteins. It also allows the valves to have some flexibility to fully open and close. So the thing that is super interesting, and maybe not so great for all of us since our life expectancies are increasing worldwide, which means we're all more and more likely to develop some of these diseases, is that in 25% of people over 65, the valve starts to become thicker over time. This is called aortic valve sclerosis. We don't quite understand why this happens. So some of it is normal wear and tear, but we don't really know what the triggers of that are. But this is already enough to increase the chances of you having a fatal cardiovascular event. And what's more concerning is that for 3% of people over 75 will develop calcific aortic valve disease. And what that means is that you start to have deposits of mineral, so your valve turns into almost bone-like. So it can obviously no longer open and close properly, and it can no longer function. How many of you, raise your hand if you know anybody who has a mechanical valve, a pig valve, a cow valve? Yeah, usually in any talk, there's at least one person. For a talk of this size, too, if it's a bigger talk, a ton of people will raise their hands. 1, because it's very common. 2, because there is no treatment for this disease. This is the only solution is surgical resection. Why do we not have a treatment for this disease? Because this is such a black box. We cannot treat a disease if we don't understand what the triggers are. And another big reason why we don't understand the disease is we no longer do because the new surgeries don't take the old valves out, but the old surgeries would take the calcified valve out, so then researchers would have access to tissues we could study. But in these early stages, you can imagine for people who have sclerosis, their valve is fully functional. So it's just really hard to obtain samples from human beings in this early stage, not to mention at this point in time, it's usually not diagnosable. Like people don't have symptoms, people are not going to the doctor. So it's just really hard to know what's happening in this early stage. And so towards the beginning of my PhD, what Kristen and I did was work with other researchers here at UW who had this swine model that's the gold standard for atherosclerosis research, so for understanding kind of similar processes in the arteries. And we thought we were going to be able to find calcified valves in these pigs, but we looked at pigs as old as 5 years old, which would be pretty old for a human, and nothing. No calcification. But what we found, which later we were like, oh, this is kind of cool. It is a really good model of early valve disease. And so one of the things we were able to find is that when you compare these pigs as they age, you can see what I was saying, right? Like in the young pigs, you have these very stratified, defined, different extracellular matrix components, elastin, glycosaminoglycans, collagen. And then as the pigs age, you see remodeling or changes in that extracellular matrix. The valve gets thicker. And the most important thing that I want you to remember is see how this teal, the blue, just keeps getting more and more and more blue. So that just means you're accumulating glycosaminoglycans. So we looked at other stuff in these pigs, and basically what we found is we found evidence of remodeling of the matrix. We knew there was inflammation, and we knew there were deposited lipoproteins. And by lipoproteins, it just means, when you go to the doctor and they're like, your bad cholesterol is too high. That was basically true for the valves of these pigs too. Okay? But what's the problem with the animal model is that this is a large animal model. So by the time we would sacrifice them, these pigs were like, they had cost a ton of money. It's not like we can go and sacrifice animals every month. One, because it's costly and ineffective, and two, it's just unethical to do work that way. So sometimes working with animals, it's a little bit like having a snapshot versus a video. So you know that things are happening, but you don't know what happened first, what was the cause of what. So this is why we felt like we as tissue engineers could make a pretty big difference. And so we knew that using our biomaterials, we had one of the materials we used as a base was gelatin, which should probably also be familiar to most people. And gelatin is basically processed collagen. So that was our base, so a material that resembles collagen, and then we would add to that material different amounts of the blue stuff, of hyaluronic acid, of chondroitin sulfate, and then we could add our valve cells to it, we could add bad cholesterol, we could add all sorts of other stuff and test hypotheses in a matter of 3 days, 5 days, with like 10 replicates. We could do it very quickly, much more quickly than what we could do with an animal model. So we could accelerate that process of testing and generating hypotheses. And I'm not gonna show you data. The point I wanna drive home is that this is where the animal, right, we started with a super big black box. This is where the animal model left us, and this is where we were at the end of my PhD. So we were able to establish a lot more mechanisms, a lot more causality. I'm gonna go through this super quick only to give you a public service announcement of really try to limit your intake of fried foods because it's not good for you. So the healthy valve somehow becomes enriched in these glycosaminoglycans. Our work was not sufficient. My lab, I think now has a good hypothesis, but at the time, our work was not enough to know why. But once they become enriched, what happens is, if you have regular bad cholesterol and modified circulating through that valve, it's fine, nothing happens. If you have oxidized bad cholesterol, which could be because you ate a lot of fried foods, maybe you're not eating a lot and you have a lot of free radicals floating through your system, and that's oxidizing that bad cholesterol, that will get stuck in that matrix because it's so hydrophilic. And then that bad cholesterol will then signal to the valve cells to activate them. And then those cells produce inflammatory molecules, and then boom, you have a cascade of events. Okay? So, and we did all of that with our tissue engineer models. That's how we elucidated all of that. Okay, if you want to read more about this is work that has been published, so you're welcome to go find it. But then end of my PhD was nearing. It was about 2015, and Kristen started being like, okay, you're gonna be a faculty member, now you have to think about postdocs. What are you gonna do for your postdoc? And I told you I love the ECM, but I've never been an obsessive scientist. Like, I'm not the person who stays up late at night thinking about, some people, I have colleagues who are like, ever since I was 9, I really wanted to study stem cell regeneration. This is my passion in life. And I have colleagues who are genuinely like that. Like that was their goal from the moment they were like in high school. And that is not me. And now I know that it's because I'm interested in a lot of things. So I'm just convinced that like if you give me enough information, eventually you'll convince me that your problem is cool and I should be working on it. So like how was I supposed to choose what to work on? I like did not know how to answer this question. And this is actually where the Colombian Badgers come in. So around the same time, we started the Colombian Badgers, several of us, and we kind of came together. It was not my idea. Other people convened the group. And so, but we were thinking we needed, this did not exist. Yesterday, it really warmed my heart to see so many talks from Latinx faculty members. When I was here, up until the Colombian Badgers, I had met none, not one. Okay, so when I was here, that was not a thing. And I had a lot of friends who were also in the PhD program who were Latinx and Colombian, and we just all kind of came together and we were like, you know what, we all know of a Colombian, we know of another, we know of another, we know of another. Why don't we all come together and like create this society to support Colombian faculty, staff, and students? And like our organization at the time had all three. And nobody wanted to be president, and because I like when nobody steps up, I have a tendency to step up. It is not a positive trait. You think it is not. It just means I get overworked. So I ended up having the honor of being the first president. And like I said, I met some of my closest friends there. I will be seeing a lot of them this weekend, be staying with one of them this weekend. But the thing that was the most impactful in terms of my career is that I started meeting the Colombian faculty on campus. And there's actually a lot of Colombian faculty on this campus. I just didn't know them. So for example, I met Tim Oswald, who's in mechanical engineering. And Tim had a ton of connections in Latin America. A lot of people in his lab were Colombian. He once gave a talk and he told us he took students from UW who were in one of his polymers classes to Colombia to look at rubber trees. My mind was blown. In my 9 years in the United States, I had never met a single Latinx faculty member in engineering at my institutions or at conferences. And he was also Colombian. My mind was blown. I also met Professor Beatriz Botero. She is an expert in contemporary Latin American studies. And from her, I learned that you could work here, but you can still stay connected to your roots through your scholarship. Like you don't have to divorce the two. And perhaps the person who's the most influential in the trajectory of how my career changed is el profe Jorge, which now sounds super confianzudo because he's now the director of the Global Health Institute on campus. So he's like a really big person on campus. And Professor Osorio studies and develops vaccines to treat viral diseases like influenza, like chikungunya, so like the types of diseases. And I think yesterday we were talking about that, right? About studying things in the tropics. And that also happens in biomedicine. That area of the world is also not always doesn't receive enough attention. At the time, 2015, 2016, if you remember, Zika was a big thing. And while meeting him during that time, he got this huge NIH grant where he could do research in Colombia. He has always had a really big intellectual and physical footprint with his lab in Colombia, and that blew my mind. So I know maybe in some of your disciplines it is more common for people who work in different parts of the world. In mine, in biomedical engineering and especially in tissue engineering, this is not common. Like it is a discipline that is dominated by countries in the global north, and I will show you data to back that up towards the end of the talk. And so to me, that was mind-blowing. Like prior to knowing the three of them, I really thought I had to split my identity. Like Ana, who works and stays here in the US, and Ana Maria, who like travels back and forth. It had never crossed my mind that you could do both things at the same time. Like it had never crossed my mind that I could try to build a career where we're actually working on problems that affect people in Colombia, that I could be like traveling back and forth to meet with collaborators in Colombia. That just like was not in the realm of my imagination. And so after meeting them, ideas started, especially after meeting Professor Osorio, ideas started percolating in my brain. So the next time that Christine asked me in 2016, I was like, I know what I want to do. I want to build tissue engineer models to study tropical infections. Did I know anything about infections? No. But I was like, I think this is a place where we can do a lot. And Kristen was like, actually, that's a brilliant idea. Nobody's doing that. Like, that will hit so well when you're on the market. Problem is, who are you going to train with? Like, nobody in our field does this. So we went, I'm going to spare you. That's another really long story of how I found my postdoc. I'm going to spare you the long story. We talked to so many people that fall, so many. I interviewed with so many people. The BMEs were like, I just don't do this work. And the infectious disease people were like, why would I hire an engineer with no expertise in infectious disease? It ended up being a mess. So very long story short, I ended up at Cornell. And so at Cornell, I ended up studying the microbiome. Did I know anything about microbes? No. Was I a microbiologist? No. But my postdoc advisor had an interest in global health. I had an interest in global health. She was new. I at least had expertise in some of the things she wanted to do in the future. I had worked with animal models. I had a ton of cell culture experience. So there were some skills I could bring to the table. Not that many, honestly, in retrospect. But I think she was also kind of desperate to hire someone. And my PhD advisor was like, the worst that can happen is you hate it and you quit. Like, a postdoc is not a PhD. You can always quit after year one. But if it works, it will be like, it could be life-changing. Like, go try it. So by now, probably a lot of you have heard about the microbiome, yeah? And its association with, like, every disease out there. Something that you might not know, and it is something that I had to confront when I started doing this work, is that this is a plot of the origin of the samples that are available in public repositories of human gut microbiome data. Okay, this was a study from last year. And most of them, if you see here in the light pink-ish, like, yeah, they come from Europe, Canada, and the United States. That's really where most of our knowledge of the microbiome comes from. To some extent, a little bit more Eastern and Southeast Asia. If we look at Latin America and the Caribbean, it's like barely a sliver, okay? So we really know that the American and European microbiome is associated with many diseases. I am very skeptical in terms of how that translates to other parts of the world. I don't know, right? Why does it matter, right? You could argue, some people argue that some parts of human biology, why does it matter? Like, human bodies are human bodies. Turns out for microbiomes, it matters a lot, because depending on what part of the world you live in, you have a very different microbiome, right? So this is just one example of why that is the case. If we look only at the order Bacteroideales, people who live in the global north, their guts are really dominated more by this type of bacteria called Bacteroides. And people who live in the global south, not universally, but for the most part, their guts are really more dominated by bacteria of the genus Prevotella. And the main thing is we don't understand how these differences affect host health or their implications for the findings that we now have. We don't know what things we're finding about the human gut microbiome are generalizable to all populations or are population specific. And so I guess this is an idea that I do obsess about. And so the kind of the big picture question for my postdoc was whether geographic differences in gut microbiota composition have an effect on susceptibility to enteric infection using kind of a gold standard model for human gut microbiome research, which are germ-free mice. So germ-free mice are mice that don't have microbes, and then you can transplant them with microbiomes from whatever part of the world. And people do studies in these mice all the time. So there's studies linking the gut microbiome to obesity, to autism, to a bunch of other stuff. So people use this model a lot. And again, I'm not going to show you data because I want to wait until the end to show you more data from what we do in the lab now. But as a summary, what we did is we took microbiota from people in the United States, in Fiji, and in Guatemala, and transplanted that into germ-free mice, right? So they became more US, Fijian-ish, and Guatemalan-ish mice. And then we would infect those mice with a bacteria called Citrobacter rodentium, which mimics an E. coli infection in the human. What we actually found was that the Guatemalan mice were the most resistant. Because I knew somebody was going to say that. So because we're in a Latinx audience, I have to caution you. When I presented this work in Guatemala, everybody was like, yes. And I was like, no, these mice are not humans. Like, these mice are really strange. I don't think it says anything yet about human biology. It's just showcasing that when we use the exact same model with microbiota from different parts of the world, we get 3 different results. Okay, so it's a cautionary tale about interpretation of data. It isn't, I'm not trying to suggest that like Latin American microbiomes are more resistant, make you more resistant to infection. Although if you want to believe that and that gets you through life, I'm not going to get in your way. We also found that if we co-house the more susceptible and most resistant mice, which were the Fiji and the Guat mice, there could be a microbiota transfer. And because of that microbiota transfer, then the susceptible mice became resistant. So we could attribute it to the microbiome. If you're interested in that, you can also go read the full paper. It's a cool paper, and it's cool to see. If you have questions, happy to chat about it later. Okay, so, but this project also taught me a lot of stuff about myself, a lot of the stuff about the way we do science, a lot of the stuff about, somebody mentioned yesterday, I think it was our first speaker, I think it was you. You mentioned how like you have been equally in Mexico and in the US, same for me, equally in Colombia and in the US, and this was a little bit eye-opening in how like Americanized my way of working was. So when I started working with our partners in Guatemala, Noel Solomons and Gabriela Montenegro Betancourt, I kind of had to relearn the way we build relationships in Latin America. Here in the US, it's like super business-based. In Latin America, no, you don't. You have to like sit down, have a cafecito, chat, like, you know, you have to like build that relationship before we can actually start doing things. Or for example, in Latin America, labs are not named after people, labs are named after themes. That's why when you introduce me, my lab has a name, because I don't want to communicate the wrong thing to my partners accidentally, because we have different cultural norms. And then I'm not going to have time to talk about this at all, but during my postdoc, I also started doing a lot of science communication. So you mentioned it briefly, and I will say, I used to say I do this all the time. Like in reality, since becoming a faculty member, I've kind of stopped doing micromartes, but it's my goal in life to like start it up again. But for a long time, I was teaching people on social media about microbiology using crochet. And that got me to be involved with a bunch of other stuff. I'm really involved with an organization called Clues de Ciencia in Colombia. There's Clues de Ciencia branches in Mexico, in Bolivia, and Brazil. I currently sit on the advisory board of the Story Collider. If you're familiar with them, it's a storytelling show. I more or less strong-armed them into starting doing shows in Spanish, because that's like the hill I will die on. That like science communication cannot be monolingual. And so, which is why they were like, well, you've asked us to do this too many times, now you have to sit on our board and help us do it. But in doing this, another thing that I discovered was that I had to relearn how to speak Spanish. I learned everything in English. Probably some of you are experiencing that right now, right? Like if somebody asks you to give a talk in Spanish, like in our native tongues, you would be like, you would freak out, right? And that's sad. It's very sad. And so in doing this, I also reconnected with my family. I could actually explain what I was doing with my friends. I felt closer. I felt more like, I felt more of a whole person at work, because I felt like I could fully communicate everything I was doing with every part of my identity. And so this is also why I'm such a big advocate for giving our students an opportunity to present their work in Spanish or in whatever native tongue they have. It is so, so, so important for your identity as a scientist and as a professional. And so now you might be wondering, I told you like a lot of different things that I do. What do we do in my lab? So this picture was taken at the end of my first year. So this was, and I've only been there 2 years and 3 months, but you'll see my lab has grown a lot. So this was the first iteration of our lab, my first 2 PhD students, Sam, who was a master's student at the time, who's now a PhD student in the lab, and our first 2 undergrads. And of course, I chose a picture that totally showcases how we're in Florida. So we kind of combine those ideas. So we use engineering tools to create physical models of disease so that we can study host microbe interactions mediated by the extracellular matrix. And for a lot of our projects, we are specifically interested in microbes or problems that are particularly relevant to people in the global south. And honestly, I often say in the global south, but I'm gonna be honest with you, I mean Latin America. Like, for now, there's only so much we can do. I really just mean Latin America. And so let me share with you a quick story well, the story is not quick, but I made it quick for this. And because we're on YouTube right now, I cannot share, and my lab is very young, so we're kind of vulnerable right now. I cannot share a lot of our preliminary data, but I can share with you the last story from my postdoc that we are continuing to explore in my lab. So it really showcases what we do now. Okay, so what do we know about inflammatory bowel disease? So anybody knows anything about IBD in the audience? Shout out some things that you might know. It is made worse by anxiety, right? Yeah, that's a great one. Anyone else? So, and inflammatory bowel disease is really a whole spectrum of diseases that includes ulcerative colitis, Crohn's disease. It is very painful. That's something that people sometimes say, gastrointestinal distress, changes in the human gut microbiome. Those are all the things people say. What a lot of people usually don't say, that is a really important hallmark of these diseases, is that my friend, the extracellular matrix, also plays a big role in these diseases. And it is a big hallmark in two ways. One, in sometimes, like in colitis patients, there is excessive breakdown of the extracellular matrix. And in other cases, like in Crohn's disease, is the opposite problem. There is too much extracellular matrix. That's why often Crohn's disease patients have to have resection surgeries, because their colons get so thick that stool cannot pass through, and it is incredibly painful. And it is because of this excess in extracellular matrix. We're starting to understand slowly that proteases, so the enzymes that break down, that could potentially break down these molecules, they have been proposed as markers of IBD. So their levels are elevated in the stool of these patients. But right now, current hypotheses point to the human cells as the main cells that are responsible for doing all of these remodeling or modifications to the extracellular matrix. So for example, immune cells or fibroblasts. A good example of a fibroblast-like cell would be like the valve cells that I talked about in the first part of my talk. But we know that bacterial pathogens can actually interact with human ECM to invade deeper into tissues. More recent work has also started to suggest that members of the microbiome can secrete proteases and do secrete proteases that are linked with the severity of these diseases. And so my hypothesis, based on my background, is that commensal or quote-unquote good bacteria can remodel ECM, and that those changes in turn, because of what I explained before, the ECM can signal to cells, is also what triggers inflammation. And so the first question always when you start with kind of a crazy hypothesis is that even possible? Like, not even in the human body, like in a test tube, can that even happen? And so that's what we did. We took a bunch of species found in the human gut microbiome. You might recognize some of them if you take probiotics, or if you look at like kefir or yogurt, right? So lactobacillus, Bifidobacterium longum, those are like probiotic species. Species that I had already mentioned earlier that are found in the human gut, Bacteroides, Prevotella, and this species has been linked to Crohn's disease, so that's why we picked it. And then we picked, right, so you can see I primed you early in my talk. Then we picked a lot of the extracellular matrix components that I've mentioned before: collagen, hyaluronic acid, chondroitin sulfate, the stuff I studied in my PhD. And then we could test for each one of those. We designed different assays, ways to measure how much they were degraded. So this is an example of one of them, and I'll walk you through the graph. A bigger bar means more degradation. Bacteria that are colored the same belong to the same genus. And the closest they are to each other, the more similar they are to one another, the closer they are on the phylogenetic tree. Over here are our probiotic strains. Over here, kind of the strains that are common. This is the one that's associated with IBD. So we could do that for a bunch of different ECM molecules. I'll just show you the summary here. Kind of our takeaway from this was that I was kind of shocked that for every single ECM component, at least one species could degrade human ECM. In most cases, more than one species could. And you can see some trends emerge. The Bacteroides species, the ones in teal, are just really good degraders. They just really like these proteins as a substrate. The probiotic strains really don't. I don't know yet what the biological meaning is. I'm just pointing out observations. And kind of some trends started to emerge. And then, of course, I think this happens less if you're saying a straight up biology department, but I'm in a biomedical engineering department, so biomedical engineers are obsessed with this question. But is it clinically relevant? Who cares that you can find all these things? Does it matter in the patient? Actually, it's even worse if you talk to IBD doctors. And I'm like, I don't know, that's the point of the research. They'll be like, but why, Anna, how do you know that it's relevant in this grant? And I'm like, that's why I'm writing the grant. So what we did is we collaborated with Dr. Randy Longman at the Weill Cornell Medical School in New York City, and he sent us samples, like microbiome samples from actual patients. So healthy patients and patients who suffer from ulcerative colitis. So then we could grow these communities into different types of media, collect the supernatant, so you like centrifuge the cells are at the bottom, and everything the cells release into the media is left in that supernatant. Took that supernatant, same assays, and what we found is that one, even for healthy patients, there is some degradation, but for the colitis patients, it's just a lot higher, and it doesn't matter which media we use to grow our samples in. And that's true across different extracellular matrix proteins. So this is collagen, this is laminin, but it was also true for hyaluronic acid, chondroitin sulfate. And so we think this and other stuff that's in that paper suggests that it could be clinically relevant. And then the last thing, sometimes we also work with mouse models and in vivo models. We can kind of engineer them too. And so the last question, we wanted to use a mouse model to explore this idea of, does the ECM degradation make inflammation worse in vivo? And so we took 3 strains that had high proteolytic activity, Ruminococcus navis, Bacteroides fragilis, and Bacteroides theta iota omicron. So you don't need to remember their names, just remember that they had high degradation in our prior experiments. And so we have our mouse model. In these mice were fed with this substance, dextran sodium sulfate, and that DSS kind of induces already inflammation. So we did a pilot study in which we tried only giving them the supernatant, and the supernatant alone is not enough to induce inflammation, so we think something else also has to happen at the same time. And then we also fed the mice every day with the supernatant from these bacteria that were very good at degrading extracellular matrix. Then we asked, does it make the inflammation worse? And so this is a disease activity index graph, which means the higher the number, the more symptoms the animal was exhibiting. Down here, this is our control, so animals that basically didn't receive any sort of treatment. In the light grays, animals that received DSS. And in the colors is animals that receives DSS plus the supernatant from the bacteria. And I know it's a lot to look at, so this is the only part of the graph that I want you to look at right now. And what you can see is that by day 6, day 7, we actually see worsening of symptoms sooner in the mice that received the bacterial supernatant than in the mice that didn't. So in the end, they all reach the same level of symptoms and same levels of inflammation, but what we think happens is that it accelerates the process of seeing inflammation. And again, so much more we need to do there. You can read that paper too. But we think this initial study highlights that bacteria or microbiome ECM interactions may play an important role, not only in IBD progression, but also just in tissue homeostasis in general in the gut. And that's exactly what some of my students are continuing to study. What role do gut microbes play in IBD, ECM remodeling? And we hope to also do this with microbiome samples, not only from Florida, but also from Colombia and from other parts of Latin America. I have another student that has gone back to the aortic valve, and so she's studying, and we actually have really cool data here that I'm happy to chat about if you want to chat about it later. We are also wondering whether gut metabolites that are in the bloodstream can activate aortic valve cells, which would then initiate changes to the extracellular matrix. And then this is kind of my passion project, because we're actually now finally working on tropical infectious diseases. So I collaborate with Peter Kima at UF, and we study visceral leishmaniasis. It's a tropical parasitic infection that we do see in places like Colombia, Brazil, in a lot of the tropics. And we're trying to understand why is it that some Leishmania parasites cause massive liver enlargement? Is that linked to extracellular matrix remodeling? We're working with him to develop models for that. And then Yeah, I have time, especially since we have the panel after. So I'm going to show you one last little vignette. And this is actually preliminary data from the lab that I can show. And this is to highlight what I was saying earlier about biomedical engineering being very dominated by people in the global north. So another of the things, so my lab, right, we have a science side, but I am broadly very interested in this idea of increasing participation in science globally and also here within the US. And so that involves, we even have projects doing research on the science of science communication. I do a lot of stuff with multilingual science communication, with art in SciComm. But we've increasingly have become obsessed, probably because we have somebody at UF who I'll introduce you to in a second who is also obsessed with this, with doing meta-analysis of the literature to see who's publishing. Who is publishing, where are they based in the world? We've just become obsessed with this idea and doing a lot of different types of fun things. So for this study, we are really interested in understanding global participation in biomaterials research in general. So we also have a problem related to microbiome science, but this field is huge, so it's just been slower because we have so many papers to parse through. So the key question here is, what is the geographical distribution of researchers publishing in biomaterial science journals? And this is work that is led mostly by an undergraduate student in my lab, Alejandro Hernandez. He's Bolivian, with a lot of help from his mentor, my PhD student and an NSF GRFP fellow in my lab, Karen Mancera Samar, who is Mexican. So this is also why I wanted to show the project, since it's two Latino students working on it. And so Karen and Alejandro have been working on this. Alejandro started work. I had the idea while Alejandro was studying abroad in Spain, and he could have found a lot of more fun things to do while studying abroad in Spain than working on this project. And so I pitched to him the idea, what if we do an analysis of who has been publishing in biomaterials in the last 5 years? And this is inspired by a lot of work in evolutionary biology and ecology that they're so far ahead from any other field in understanding this. So what Alejandro did was go to the SciMago journal rankings. He took the top 30 journals that SciMago themselves classify as publishing biomaterials work. So these are the 30 journals. Probably if you're not a biomedical engineer in the room, the names don't matter to you, but we've learned that in the biomedical engineering conferences, people are, but what are the journals? Are you really choosing the top journals? And so we put up the names so that people know. And out of the 30, we grouped the 14, because some of those journals publish other types of material science, not just biomaterials. So for this, and that's what Alejandro is doing now. For the data I'm going to show you, we only selected the 14 that only publish research on biomaterials, so they're here. And they are indeed the most famous journals in our field, like Acta Biomaterialia, Biomaterials. Those are the most famous journals in our field, the highest impact journals. We went to Web of Science and retrieved the records for the publications from the past 5 years. Oh no, it's missing one thing. Okay, yeah. I'm missing a slide, so then we used the RefSplit-R package that was developed by Emilio Bruna, who is this person that is obsessed with this idea of meta-analysis, and Emilio is based at the University of Florida as well, so I'm really sad the slide with his picture went away. So we use this R package. We can extract the individual publications. In total, we're looking at 22,000 papers. Then, using his package, we can extract the authors from each paper. In total, that's 161,000 authors. Keep that number in mind, because that's going to be our denominator. So a lot of people, 161,000. And then we can geolocate them based on the affiliation that's reported. So keep in mind, it's because a lot of people ask, oh, how do you know where the people are from? We don't. It's where the people were based when the paper was published, right? So that's an important distinction. These 114 countries are represented in that data, okay, of where the authors were based. These are the top 5 countries with the most authors. I don't think these results are particularly shocking. That is these 5 countries. China and the United States, you can see the gap here, dominate by a lot, right? And then there's South Korea, Germany, and India. If we put this on a heat map, and I know yellow says 500, but for a lot of countries in Latin America, that's fewer than 50 authors out of 160,000. It's sad. And for some countries, like Colombia, it was something like 20. And I refuse to believe that, because I know a lot more people than that working on biomaterials in Colombia. It is not, because people are often like, oh, but what if it's there aren't enough researchers? And I'm like, I don't think that's true. right? Like, yes, we were expecting to see underrepresentation, but not to that extent. And I don't have time to show you, but if we look at representation in editorial boards, it is even worse. In all of the editorial boards, there are, in total, if you add them up, there are 2 researchers from Latin America. Two people. And so if it weren't from Brazil, it would just be so sad. If we group those by regions of the world, according to the World Bank, you can see for sub-Saharan Africa, it is, yeah, really bad. But Latin America and the Caribbean and sub-Saharan Africa are particularly underrepresented in our field in general. So yeah, these data, I like presenting it. I think it's very impactful, but it also makes me deeply sad to look at it. I knew we were going to find disparities. I don't think I ever anticipated that it was going to be as bad as it is. So one of the things we're doing now is looking at the rest of the journals that also publish other types of materials research so that we can understand if this is specific to the subfield of biomaterials or if it's all of material science. So we're trying to explore a bunch of things. If you have colleagues who work on biomaterials in other parts of the world, help me get in touch with them, because we're trying to build a network so that they can tell us what they think is stopping them from being able to participate. I strongly suggest the editorial board plays a role, an important role, probably publication fees, so we're also doing some analysis on that. The other thing we can do, and I apologize, this graph is awful. We haven't yet figured out how to plot this in a better way. So this is what happens when you plot the collaboration networks in just one of the journals, okay? And so, but the main thing I want you to notice is that a lot of the collaborations happen north-north or north-south, and there's very little collaboration, and Emilio has also found this in his work, very little south-south. So not as many collaborations between, say, people in Latin America and people in Africa. And we think that in order to increase global participation in science, that needs to happen more. Like, it cannot be that like you need a partner in the global north. Obviously, right now, we all know you do to get published in the right places, right? But like, if we want to change that, there has to be a way for those of us who work in this part of the world to help figure that out. And I don't know what the solution is, but this is just some of the things that come up in our data that are also coming up in other people's data. Oh, and yeah, you can see here, this is a small snapshot from the editorial boards, and you can see for Latin America and the Caribbean too, sub-Saharan Africa too. The rest of the world, super overrepresented. So yeah, so with that, I'll close by saying that I hope I was able to showcase how all the different things I've done in my life kind of get us to what we do in my lab now, but that is not the actual point of my talk. The point of my talk is that none of that would have happened if it weren't for the Colombian Badgers and for these 3 people. So that my hope for you is that I hope this community and whatever you can find here at UW can equally inspire you, can open your eyes to different ways of doing things, different ways of working, can inspire you to explore new ideas. This isn't even all the people in my lab, but my lab has grown. We now have 4 PhD students, several undergrads. We're growing a little list of collaborators. The work here at Wisconsin was, I had an American Heart Association fellowship at the time when I was a postdoc. I was a Cornell Presidential Fellow. My 2 domestic students are funded through the NSF GRFP program. And another thing I did not have time to talk about was the Latinx and BME community. And we are actually hosting our first in-person symposium next week, which is really exciting. And with that, oh my gosh, I took a whole hour. I'm sorry. I'm happy to answer questions later.
We have maybe time, 1 minute for questions, but then Ana Maria is also going to be part of the forum next, so we can also ask questions then. But if anyone has a question right now that they're afraid they're going to forget for later. Yes?
I had a question, but it's about your last part. So when you show that there's like very little representation of countries in Latin America in bioengineering, so how much do you think that is also due because the lack of funding there, right? So and I don't know anything about like your specific field there, but how can we help, or what can we do? And I don't expect you to have all the solutions to make sure they also have the resources to do competitive science, because there's like bright people working in problems there. They just don't have the resources to make it to a like top tier journal.
Right, exactly. And I do think that's a huge factor. Oh, I'm so sorry. So the question was about do we think that the fact that it's really hard to find funding, that plays a role in being able to do research as competitive for these tiers of publications? So yeah, I think that's a big factor. I also think even if you were doing competitive science, the cost of publication is also a major roadblock. And a lot of these journals do offer kind of tiered pricing, but once you convert it to the money for the country, it's still too much money. So I think that's another thing journals could consider. It's like prorating the rates better, right? So one of the analyses we want to do is compare, for example, the cost of publishing an article compared to like a graduate student salary. Because like in the US, like it's just the ratios would be completely different if we look at other parts of the world. So yeah, I think some of the things are not addressable easily, but there are other things that journals can do, right? And so, but yeah, it's a huge question to tackle. I do think that like you have to understand the problem, the magnitude of the problem first before you can address it.
Hi, thank you for the talk. So some of the themes that you covered, I think speak to kind of are at odds with some of the political environment. I'm a native Floridian. I grew up in Miami, was a history teacher there, led DEI there. And I think in science, there's sort of this idea that, well, the impact is felt most in the humanities or social sciences. And from your perspective, especially just given the connections that I think you're drawing between your work and society, what do you see as the risks, as the dangers relative to science, just given the environment?
I will not answer that with live YouTube streaming. Sorry. I happy to answer that individually later.
Okay, let's thank Anna for now, and then we're gonna set up for the forum. Okay, no one can hear me. So thank you, Anna, so much. We're gonna set up for the forum, which is our last activity. We're gonna probably need maybe like 3 minutes if people wanna take a coffee or something else to eat. And then if those that are gonna be in the forum, they can just come start sitting here, please. Okay. Okay, let's start taking our places back. Okay, well, thank you. This is the last event for our symposium. I just want to thank again everybody for participating, the speakers for agreeing to present and also to be part of this symposium. And I want to also add thank yous to Wisely, Wade, and Cal's for the support. So, as the last event, I thought that we could have a panel where people just sharing their experience in Latinx community, both under, whatever they want to share from their personal life, but also in their work in STEM as a scientist. And I will have 4 questions that I plan to get us started, but then we're gonna open some polls that people can ask questions via a link or also here in the room if they want to just say them out loud. Okay, so we're just gonna start with like a brief intro and just a short bio. If you can just tell us your name, your department, where you were born, what year, your favorite pop artist. No, just kidding. Just whatever you want to share with us. We're gonna start here with you, Angel.
All right, good morning, everybody. I'm Angel Adames Corraliza. I'm originally from Puerto Rico. I grew up in San Sebastian, which is in the northwest of the island. I was always really interested in our planet, the Earth, but like my interest in my field, which is tropical meteorology, didn't really kind of blossom until like Hurricane George hit the island in 1998. And after that, my family really kind of like supported the idea of me doing a career in atmospheric sciences. However, like that isn't a degree that's offered at the University of Puerto Rico. So what I decided to do was so I ended up getting a degree in physics at University of Puerto Rico, Mayagüez, which is where a lot of the students in my discipline tend to come from, and also Rio Piedras is the other one. And then after that, I had a couple of important experiences, summer internship experience that really defined my life, that we can talk about later on, that led me to go to grad school, made the decision for me to go to grad school where I got a PhD. And then I've been moving around like a hot potato, and now I'm a faculty here at the University of Wisconsin in Madison. Quirks about me, that maybe are useful. I like ice cream. I have a really crazy sweet tooth, and I've been really craving ice cream since yesterday's talk about the waffle cone. So there's that, and I have other things that I do outside of my job to keep me sane, including running, biking, and other kinds of sports. So yeah, that's my bio.
My name is Laura Hernandez. I grew up in El Paso, Texas, and I had no clue what I wanted to do. I swam in college, and I heard that you could do biology for animals, which really was more of my interest. Wasn't into microbes or worms or any of those things, or really plants either. And I ended up in animal science, and it allowed me kind of to focus on that. I had no clue what I wanted to do, and my dad suggested maybe like get a master's degree and you could teach high school biology. So I was like, sounds good. I could do that. I took the GRE, the LSAT, and the MCAT. That's how unsure I was. And so he suggested I do that first. And there I learned that you could do science for a living without like the pressures of a person's life or death, and the pressures of dealing with people when their animals are sick. And so I ended up doing the master's degree and had an advisor who said, like, you're really good at this. And I said, what do you do next? And he said, you get a PhD. So I did my PhD at the University of Arizona, and I was always really interested as an athlete, like, how the body can shift states and do these really cool things that they don't do when we're just kind of sitting around. So I ended up in the lab of a professor who did stress physiology, and he had 2 projects, environmental stress and lactation stress, and he had funding to do lactation stress. So that's where I ended up. And I then became obsessed with what females do when they're making milk and how they can actually stay alive, and all the things the body does to deal with that. And so did a postdoc. I've worked with cows, I've worked with mice, I work with humans. And really my passion is maternal health and making sure they get support during that period. And kind of I'm at the intersection of like depression in humans and how that affects their physiology and offspring. And then I work on a lot of stuff with calcium metabolism in dairy cows. So I really just like mammals. I really want a marsupial. That's my quirk. And my children think that's hysterical, but they love animals too. And I'm in animal and dairy sciences, and I have an affiliate appointment in OB-GYN as well. I do a lot of things. I'm the chair of endocrine and reproductive physiology grad program, and I just got it. I'm now co-director of a program I'm super passionate about for post-baccalaureates that is largely focused on underrepresented groups. And that's been amazing. So I've gotten to do a lot of great things for reasons that are really passionate to me. And I run to stay sane. And yeah, I think that's about it. I've been here for 12 years. Madison's an interesting place. I miss the Southwest, El Paso. You want to talk about growing up on a border town? Sometimes I don't know where I belong. Am I like Mexican American? Am I here? It's a strange place to be, but that's me.
I don't think you need to hear this from me.
Are you sure, Anna?
I've said everything I needed
Okay.
to say.
Okay. Well, then my name is Daniel. I am a faculty here at the Wisconsin Institute for Discovery and the Biostats Department. And I am originally from Mexico City, where I knew since I was 9 years old that I loved ice cream and I wanted to investigate more about cones.
I can.
Correct answer.
Correct answer to ice cream is my favorite food. You chose well.
Well, I seriously, I mean this, don't listen to Victor's talk about the impact of the dairy industry, because then ice
Okay.
cream will not be the same to you anymore.
Okay.
So avoid him at all costs. I don't know where he is, but anyway. So I always liked math since I was a kid, and I guess, one thing led to another. I also at some point didn't really know what I wanted to do. I started studying industrial engineering, then switched to computer engineering, then to another type of engineering, and then ended up in here. Then I came here for grad school in electrical engineering, where I don't know anything about electrical anything. I only know math, and yeah. So now that's what I do. I love it, and on the hobbies side, I would say that we should all get together, because I also like running, biking, ice cream. I also like all sorts of things. So
Combine them all today.
yeah.
Let's go running, biking, and then get ice cream.
yeah.
Actually doing that. So everybody here, we're gonna do the bike ride. You all got the email about that? No? Okay. Just kidding.
That's all I got.
Thank you. Okay, so we're gonna start with one of the serious questions. Just have you faced any challenges related to the gender and/or race or ethnicity, or actually anything about your identity in your career, and how have you overcome them?
So I like to begin these kind of conversations by just being honest and upfront about them, like what your experience really is, right? And there's two sides of my story, right? Like, I think it's important for me to start off by recognizing the privileges that I have, because I am male, I am cisgender, and as far as Latinx people go, I'm pretty white as well. So there's a lot of things that like a lot of my Latinx friends experience that I don't experience. However, I will mention that that doesn't mean that I haven't had any experiences, which I think kind of in itself speaks a lot. I come from low income. Uh, I didn't know what the right way to become a scientist was, if there is one, uh, and what the things that I needed to know were. Many of you know now that there's like this hidden curriculum for grad school, things that you kind of need to know that nobody ever tells you. If you don't have anybody in your family that has an education, like how are you going to know these things, you know? And if you come from a university where you don't have mentors, like, are you going to find out these things, you know? And, you know, I'm a pretty chatty person now, but like I was a lot more shy when I was a student. I was like afraid. So I never really spoke up about these things. But, you know, coming from Puerto Rico, a lot of people are like me, you know, when I study. So like I never really felt like I was super different until I went to the States and started grad school. And then I realized that like something wasn't quite right. You know, and now I know that these are called microaggressions. So, you know, students, you know, often mean well, you know, and sometimes they know what that they need to be allies and stuff, but that doesn't mean that they practice what they preach, right? And I think that's pretty common in grad school. And sometimes they'll make comments like, oh, you know, are there paved roads in Puerto Rico, you know, like, or like, uh, you're saying something and then somebody was like, oh wow, your English is really good, you know? And I'm like, what's that supposed to mean, you know? Like, is it not supposed to be good, you know? Is it supposed to be bad? There's way more than that, right? Like, I really worked hard to not get an accent. I still have an accent. It's not as noticeable. One of the reasons was because when I went to grad school, I had a much thicker accent and people would be correcting me all the time, because I would roll the R constantly, because that's what I do, right? That's what I do. My native language is Spanish, and in Spanish, the Y and the J, you know, they don't sound like the Y in English. It's just a little bit different, but enough to kind of irk my classmates that it's like, it's not pronounced this way. And it frustrated me because like there were British, French people, German people in my department, and I never heard my colleagues correct them. They have accents too, you know, so why is the Hispanic accent getting corrected but not the other accents, right? And there was like all these things, you know, that like people expected me to know, but also expected me to do, right? Like people were like, oh, why don't you have a bike? Oh, why don't you have a car? Oh, why don't you, you know, like know these things about these classes and stuff? Like, why didn't you take these classes ever? And I'm like, why am I supposed to like have a car? Like, cause I'm broke, you know? And like all the money that I had from college that I had saved up from my scholarships, cause my family didn't have any money. So I had to like raise all the money from doing REUs and trying to get scholarships in order to be able to move to the US, cause moving to the US is expensive from Puerto Rico. Like, it's not like you can bring a bed and you can bring it in any of these things, right? You have to kind of like just leave everything and bring a backpack full of books and then like figure everything out, right? And so I ended up like saving like $5,000, and then by the time I had moved to Seattle, because I went to University of Washington, the other school with a W, and so by the time I had finished, I had no money left, you know, because it cost me like roughly $5,000 to actually go to the surplus store and get like a desk and get like a bed, you know, and get like all these things that I really needed that a lot of my classmates didn't have to do because they were from the States, and all they needed to do was to get a U-Haul and throw all their things. And some of them had their parents give them a car, right? And I didn't have a car. And there was this constant kind of like low-key judgment. Because I was from Puerto Rico, I had an accent and I was broke. And so, you know, this is kind of like a situation where like individually, maybe those things are not a big deal, but it's more like a death by 1,000 cuts, you know, and slowly I felt more and more alienated by my class. They also did stop inviting me to things because I really don't care about American football, you know, like like I actually didn't even understand it because in Puerto Rico nobody, like you don't see American football. You see Sábado Gigante. That's what people do. Don Francisco is people watch Don Francisco. Like that's 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 Saturday night, right? Like and so that's kind of like the thing, right? Like, but then people just start judging you as like, oh, he's just not interested. He's weird. He's different. Instead of being like, maybe he just has a different culture. Maybe he just likes different things. But because you're the only minority in that group, because that's the definition of minority, right? Then you get ousted, And I know that my experience was not the only experience. That almost everybody I knew from Puerto Rico, almost everybody I had made friends later with somebody from Mexico, and he had the same experience, right? And I think that this is a big part of the story of being Latinx or being any minority, right? That you get ousted by default because you're not like the majority. And ultimately, we kind of like to have our group of belonging, right? And if you don't have that, then you're kind of screwed, And I don't want to delay any more time, right? Because you have 3 more people that are going to say things. I can talk afterwards, but like, I will end by saying that like, I never quite realized the deep depression that I felt in, and that I really needed help, and I had nobody to get help with. And I almost quit grad school on multiple occasions, and I didn't. And I did finish, which is great, right? But like, what was the cost, Like, and I think that my life since grad school has been this constant journey of finding myself and trying to repair that damage and make myself a better person because of that. And I think that like, while grad school is hard for everybody, it's especially hard for those that don't identify with the majority.
Wow, to follow that.
Yeah.
I'd say by starting with my privilege was I was born on this side of the border. And I don't know if any of you have been to a place like El Paso or to El Paso. Many people haven't. But I grew up with a mixed identity to begin with. My dad was first generation, and he was very adamant. We only spoke English at home, and we had to get no accent because he worked very hard to get rid of his. And my grandparents had a 3rd grade education, and my grandfather essentially taught himself to be an engineer. And every Saturday night we watched Sábado Gigante together. And I learned Spanish with my grandparents watching that, and telenovelas with my abuela. And so I was always kind of in the middle. We didn't speak it at home, but I spoke it with my grandparents, and it was never good enough. Where I was from. I wasn't from Mexico. I didn't speak good enough Spanish. I didn't listen fast enough, speak it fast enough. And my mother is a very tall white German lady. It's a good thing she's not watching this because I said it. And it was a real conflict because my dad had been beaten down so much. He joined ROTC so he could pay for school and went to Vietnam because of it and died because of it from cancer. So it was very hard growing up because I was treated as an other and by a group that I more identified with than the other. And I went to Iowa for undergrad. I really didn't want to go there, but my dad said I should go there, so I did. And talk about microaggressions. There were macroaggressions. They all assumed I was a drug dealer. I must be. I'm from El Paso. That's where the drugs come from. What is it like to grow up? Are like these people invading all the time? And I'm like, I don't understand what you're talking about. And I hated it. And I left after my 3rd year and went through a very deep depression. Went home and I finished there in New Mexico State and did my master's there. And getting more back to my roots and being where I felt like I could be who I was helped. I was very terrified to leave again. Arizona was more similar to where I was from, so that was okay. But had really bad experiences for, again, being like a border rat. And I could just never fit. And sometimes I still don't feel like I fit, because when people come from other countries, they find their community that way. But I'm not from those countries. But I'm also not fully accepted either, because I'm from a border town. And that is a really hard space to be in some days, because I don't know where my community is. And, you know, here, really smart people come here. And many of the professors are from University of Washington. They did her PhD, or Harvard or MIT. And just the other day, I was sitting in a grad directors meeting, and we were talking about stipends and such. And I was like, I am blue collar with these folks. New Mexico State, University of Arizona. I went to Yale. Congratulations. So I still don't feel like I fit. And I'm in a weird space with my research, being biomedical and agricultural. And let me just tell you that there are not a lot of people that look like me that are in the field that work with dairy cows. I get asked to do a lot of things because I check a lot of boxes, and I do it because I care. And it makes me really mad that, you know, there are all these things that get said. And, you know, I've been in meetings where they're like, well, I don't really care. I'm colorblind. And I'm like, oh, don't say it. And like, it makes me uncomfortable. And I'm like, I've been uncomfortable my whole life. Congratulations. Welcome. So that's really hard. When I interviewed for this job and they called me and they said I got it, I started crying because I thought they picked me because I checked like 3 boxes compared to the other people they interviewed. And my PhD advisor was like, Laura, you got it because you're qualified to do it, and the most qualified. And I still don't ever feel like I am. And it's hard, you know. I think it's why it makes me an empathetic mentor. I'm a huge empath. My daughter is even worse, and it's very terrifying to see yourself grow up in a miniature person. And she always asks me why I can roll my R's and she can't. And I was like, because that's the way it is. And so I've worked really hard to like make my 2 kids aware of my culture. I think it's really important because sometimes I feel like being in Madison, I'm afraid that my culture experience will die for them, and I don't want that. But I've been here for 12 years, and I still don't feel like I fit, for whatever that's worth. I was sad I didn't see Natalia. She's like my superhero. I often run into these things with her, and I feel like if I can do those things, like I can make an impact. And I've been really active in DEI in CALS. And I'll say they asked me to join EDC like the day after I got tenure. Again, the boxes. Wow. My chair was not happy when they asked me to do that. I was like, I don't know. They asked me, so I said yes. I didn't. But in the end, like I was on CALS EDC for 6 years. We did a lot of important things. We got them to hire Lewis. Yes. We have a DEI office now, Rosie. And that was very inspiring for me that like we're doing things to make changes. So it like the same for someone who's in this cross weird space, it's hard for me to find it as well. But my privilege is I was born here. And that just being a mile north of the border gave me a lot of things that, you know, aren't as, that are much easier. So, yeah.
It's kind of hard to follow both of you. I will do my best. So, well, you heard some of the things already, but as you were talking, I was reflecting. I think I got really lucky that the first place I lived in the US was Texas, and I didn't know anything. Like, it's not like I picked it because I was like, oh, this will be a great place to live in. No, I was just like, it happened to be the one school where somebody else from my high school had applied. He was like, there's BME here, apply. I applied, I got in, didn't even know it was hard to get in. So I just got lucky, and it's like, it was a privilege that I got to go there. And it was a really great place to live as a first place to live in the US. One, because Austin is an awesome city, but two, because like UT Austin is now a Hispanic serving institution. So like, I wasn't even the only person of Colombian heritage in my classes. So there were multiple of us. So I was never confronted with that idea that like I was different because I was Latina. In fact, I was never in the Society of Hispanic Professional Engineers because I never understood why one would need to be in one. Right? Because I was like, but why do I need to like self-segregate? And then later I realized I was just me not understanding a lot of things about the US context, and like having grown up in Colombia too, to speak of the privilege of that versus here. It's very different when you grow up in an environment where all the doctors, all the engineers, all the like everybody is Latino, so like you don't have that perception. But it was interesting how over the years you start to self-limit yourself because your experience, right? So from Texas I moved here, and here I had a wonderful experience in grad school, like I said. But even then, you like all of a sudden you're like, oh wow. And then I moved to Cornell, which was even more oh wow. So then you start being like, can I do this? Like there were before that has improved a lot since I became faculty member, because a lot of us have been hired. Prior to 2019, there were fewer than 10 in the whole country Latina faculty members in biomedical engineering. Fewer than 10. So therefore, it was no surprise that I had never met one when I saw the statistics. And so you start to wonder, like, can I do this? I don't know. The people who look like me, like nobody is doing it. I don't know if I can do it. So it kind of gets totally in your head. And like, if you think about it, like I grew up with parents who were engineering faculty. Like if I couldn't do it, then who could? Like it just makes no logical sense that I should have doubted myself. But that's what happens when you keep moving in spaces where you are more and more minoritized, and like you don't see people who look like you. That's just what can happen to your brain. And so then my solution, because you talked about solutions, so my solution with one of my friends was to co-found Latinx in BME. And actually, to speak about something you just said, something that was really important to us in Latinx and BME is that we were never going to police who call themselves Latinx. So we like have no requirements. Well, actually, we do have one, and that is that people from Spain can come to our events but are technically not part of our organization. And it is a really hard line that we drew. And we've had fights, actual fights with colleagues, Spanish colleagues who want to be part of our group. And we're like, you can come, you can support us, but I'm sorry, no, this group is for Latin Americans, people of any Latin American heritage. Heck, I guess you could also be an American who lived in Latin America for a really long time. We will embrace you. But like, come on, like, anyways, that's our only hard line, because it's really important to us that we are very conscious, and a lot of people in our community don't speak Spanish really well. My co-founder, so I was not born in the US, my co-founder is Colombo-American, and while he does speak Spanish, he brings a completely different perspective from what I do. If you look at studies, at least in engineering, most of the Latinx faculty in engineering in the United States was not born in the United States. So there is a severe underrepresentation, an even worse one from the stats you presented, if we look at Latinx faculty that were born and raised in the US. So I do think that's also something we need to think about. And then I do want to talk about gender. So I'm in engineering. I also was very fortunate that, like you saw, all my mentors have been women. I work in a department where it's 52% women, 23% underrepresented people in faculty roles. So like my department is really unique. I'm the only Latina, but I'm not the only Latinx faculty member. We have 6 Black faculty members. It's just like a very unique place to live. So I guess to kind of get at your question, I think it's one thing what you see in the news about Florida, and it's another thing with the actual experience of working there. So like 22% of my students are Latinx. My TAs, I talk to them in Spanish. A lot of students I talk to in Spanish. So it's actually like a really comfortable environment to work in. And also, it's really nice that in our department, like, you're not necessarily always the go-to diversity person because you actually have choices. And same in our college. Our college is actually the most diverse in the country, including HBCUs, I think, for Black faculty in engineering. So there is just like a little bit different. And so that has been a really nice experience. But what I wanted to say about gender, I have never experienced so many gender issues until I became a faculty member, but not from colleagues. And I feel like we don't talk enough about these in panels. It's from students. It's all from students. So I've had students, I co-taught my first semester, and the students would call me Miss and my co-instructor Doctor, undergrads. I've had grad students who wrote, even though my evaluations were great, not so great things on my, like accuse me of some really weird things. And then students have such higher expectations of women mentors compared to male mentors. Like, it just feels like you're supposed to be all these things, you can never make a mistake. And I don't think they hold male mentors to the same standard. So to me, it actually has been really shocking. Like, it has been shocking to me how much gender discrimination I feel like I experience at the hand of students, not of my colleagues, of the students. And I think that when I was a student, I never reflected on that. How was I behaving toward my faculty members? Was I contributing, right? Because when you're a student and a trainee, you think a lot about the people from above suck, all of these things are happening. And we don't maybe spend enough time thinking about how do we contribute to a problem. And yeah, for me, it has been students. So I just wanted to highlight that because I don't think we talk about that enough.
Well, okay, I've had some of all of that, except obviously the gender issue. My story is kind of similar, kind of very different. Well, my lucky break, in a way, was that I was born in Mexico City, and it's very homogeneous, and so everybody there, looks kind of like you, and you see doctors, engineers, same thing, right? And so you don't grow up with this, oh, I look this way, I can only do this type of job, which is something that seems to be very prevalent here in the US. So that was my lucky break. So I never really had a mindset of, oh, I cannot do that. And then I had the lucky break that, well, I applied to UW. And here is where I think, just a little bit of help can go a long way. Like Angel, I didn't have any money, and I didn't even have the $5,000. 5,000. I don't think I had ever seen 5,000 pesos, let alone $5,000 together, right? So when I applied, I got in, but I didn't have no money to come. But I had the great luck that somebody here at UW was very helpful, and I will ever be thankful to him. His name is Mark Alley, and he really supported my application, and he told me, yeah, well, we will give you all this funding, we will help you come out. And just that tiny, it was not a tiny little bit of help, right? But just that effort on his side, just to say, yeah, okay, I'll vouch for this, I'll give you this help, that made the difference, right? Without that help, I wouldn't have been able to just afford coming here. And I think that if you are on that side of, yeah, I could do a little bit of, just, yeah, okay, sign this form that says, yeah, we'll give you some of that department money. It can go a really, really long way. And I think people here, if you forget a little bit about where they come from, people are just people, right? I mean, if you get a chance of accepting a graduate student from wherever they are, they just take a chance. I mean, maybe you will get lucky, you will get a good student, maybe like these people, maybe you won't. But chances are the same. I mean, just because they are going to be from somewhere in Latin America doesn't mean they're going to be dumber or anything. I mean, they may be just as good as any other student. So, yeah, I mean, my story is that I think I was very, very lucky that way. And then when I got here, I think at UW, I was never really aware of any sort of discrimination towards me. I was lucky that way as well. My department was not very homogeneous. I guess that was the lucky break. There were not a lot of Latinx students, but there were lots of everything else. And luckily my accent wasn't necessarily the worst, you know. So nobody could pick on me on that, I guess, or maybe if they did, I didn't even notice. But things I feel that have gotten a little worse in the later years, especially after, you know, some recent political events, that now I don't even know if I am just overly sensitive or I am just more aware of these issues, things that normally people would tell me, and I would be like, I guess that guy is crazy. Why is he saying something like that? But now it's always in the, you know, in the back of my mind, like, is that a racist comment? Am I being discriminated? Am I? And so maybe it is also me being oversensitive. I have no idea, but it has gotten a little worse.
Okay, thank you for sharing that. I want to switch to a little maybe, you know, positive, in the sense that if you could share any experiences where you felt very proud of your Latinx heritage, or where you think it gave you any sort of advantage. The reason why some of the background for this question is that one time students reached out to me and said, oh, do you think by being Latina, you know, I mean, is there something that you are really proud of? I'm like, I'm 100% proud of being Latina. We are fearless, you know, and like very resilient, you know, I felt really proud. So I was thinking, I'm sure there's something, or maybe if you can think of some moment that you wanted to share with us.
Yeah, so three things, and I'll try to be a little bit on the briefer side. So one of them is, you know, a lot of Latin America is in the tropics. I grew up in Puerto Rico. I lived the weather in the tropics, and given that that's what I study, I feel like it's pretty easy for me to communicate with the Latino population, both in the U.S., because a lot of the Latino population in the U.S. is in the U.S., which is subtropical, experiences a lot of tropical weather. So I feel that because of my heritage and growing up in Puerto Rico, it gives me an advantage in which I can like more easily communicate and relate to people and talk about the weather that affects them. And I think that that's been a significant advantage. And I'll give an example of that. In 2017, Puerto Rico got hit by Hurricane Maria. It was the most worst hurricane that the island has faced, you know, in a century at least. But definitely the most damaging. Prior to Hurricane Maria, I had started doing more science communication in Facebook and Twitter in Spanish and in English too. And, you know, I think that there is sometimes that we recognize it, but we probably don't value it enough, especially in academia, how much of an impact being relatable is to certain people, populations. And I started talking about the forecast of Hurricane Maria, and early on it was very clear that this was going to be a really bad event. And a lot of people wouldn't believe the news because the news tends to sensationalize everything. But when I posted it on my account, people were like, this person has a PhD in this field, and they're saying that this is coming and it's going to be devastating. Like this is the real deal. And people prepared for it. Even I underestimated how much valuable I was going to be. I just didn't know until somebody called me on the phone afterwards and told me, like, if you didn't tell me that I had to leave my house because it was going to flood from the river, I would have not done that, and I would have drowned, you know? And that's tough to hear, but it's also important because you don't know what you can do, right? Especially like to these communities, to people who don't know how things like just a simple weather event, right? How that impacts them, and how them having somebody that they can trust, how much that means to them, you know? And I think that that was the moment. I've been active, you know, I go in periods of inactivity because this job kind of sucks sometimes, honestly. But I think that that was probably one of my proudest moments, you know, even though at that moment it was really intense and really difficult. But like, and afterwards it's like, wow, you know, you actually helped people. in a related sense, the other one that I think I'm really proud of is, and I think this is one where a lot of us here relate to, is that there aren't a lot of like Latinx faculty professors, and one of the things that I'm proudest of is that, like, to me, diversity brings diversity, and just by the fact that you have a Latinx faculty in your department, our department has 3, which is not a lot, but it's actually one of the highest numbers in the nation for in our field, atmospheric and oceanic sciences. It just tells you how bad things are. And I get a lot of emails from students from Latin American backgrounds, both from like U.S., Latin American, like Latinos in the U.S., also people from Latin America. I've talked to colleagues. My colleagues do not get any of those emails. I get a lot of them, almost all of them. There's the other Latinx faculty do too. I think there is an unspoken truth about like you being somebody that they can relate to, being part of the draw, and I think that's important too, right? Like when you're a grad student, you kind of want to have an advisor that you can relate to and have a good relationship with, because it's going to be really hard if you don't, right? It's 5-6 years of your life that you're going to spend with that person, like not really like, like this person is going to be like one of the most important people in your life during that time. And, like that matters a lot, I have probably the most I have the most amount of Latin American students probably of any group, probably in this nation, right? Like I have more than half of my group at one time was like Hispanic. I have Puerto Ricans, Peruvians. I had somebody from Mexico, also somebody that was like Mexican American. I've had like literally Latinos from all over the place, and to me that is the most proudest moment. And I know that that's partly because I'm Latin American as well. And to me, like being able to be that mentor and being somebody that's relatable, somebody who can empathize with them and understand their suffering, like these things are happening to me, and it's like I know it's hard, but we are here to help you, and even though sometimes it's really hurtful for me too to hear their stories, like I know that I am contributing to change that so we can have a future where there's more of us, not just more Latinos, but more of like every, like ethnicity, culture, race, or any kind of view. And so that these things that happen to us as people and that all our students are happening, they happen less in the future until we get to a moment that these things don't happen anymore, and that everybody gets to have an equal voice in the table, because we don't have that right now. And being able to contribute as little as I can to like this change, to me, is the proudest thing I have as a Latino, especially a Latino professor.
I'd say, like, following that, the mentorship aspect, like some of the traumas I experienced, as an undergrad, but from a professor too, as a graduate student, that almost made me quit several times. And it's really quite shocking I survived even now. And like the trauma of having to see that person sometimes at meetings and such is awful. But I had a really good mentor. And I feel like those experiences have made me in that way a good mentor. And I have a really deep passion for graduate students and, post-bacs and undergrads finding who they are in a safe space. And that's been my priority. And like you, like I right now I have like 3 Brazilians in my lab, and eventually they laugh. They tell me I'm going to start speaking Portuguese, but one of them says I shouldn't because like hardly anyone speaks Portuguese. So, and I have a Hmong student, and I always tend to have lots of females. Occasionally we get a male, and he laughs, the one we have now, about being in the lab full of females. But I found that that's where I could have an impact. And when I get asked to do these things as the, box checker, over time I've gotten more vocal about it and really wanting to change the culture in places, because the boards of journals and the boards of societies should reflect the graduate student population. And the graduate student population is very diverse. very, very diverse. And I think that's where I've tried to work the hardest. Equal opportunity for graduate students, support for graduate students. And that's where I've tried like very hard to have an impact, so that they don't encounter events like I did with a particular faculty member. So I feel like I've made a lot of headway that way. I think I have a fairly good lab to be in. Most of my students say that, so they tell me to stop having imposter syndrome about it, so I try. But that's where I feel like I've had a lot of impact. And, if I don't end up discovering something super awesome, but like women can have their babies and have a breastfeeding support system and have health care, and cows can be healthy and all mothers can be healthy. That's, I think, is the forgotten link. You notice I keep going back to that. But I feel like even from a healthcare, option, we don't put in a lot of money to support women. And in their research, it's all really focused on what happens to the baby. And I love babies. Like, I have 2 kids. I'd have 10, except that's against my husband's wishes. So we don't have 10. He says I'm too busy anyway. But I that's where my research, I feel like, is having an impact. And I hope to keep doing that to support, women. And I think, like you said, people see me, they see my name, and they're like, I can do that. There's someone else who looks like me. She's doing it. And if there's that one person you see sometimes, like, that will completely change the trajectory of your life. And I've had a couple of people like that in my life in times that they had no idea I needed it. And if I can do that for some people, then I feel like I've made change in the world. And that's really where I try to fight for everyone who feels like they can't find that. So that's what makes me happy, not writing grants. That does not make me happy. There's a lot of rejection in science. Sometimes the skin just has to keep getting thicker with the extracellular matrix to deal with it. That part drives me nuts, but it really is the mentorship. I love graduate students. That's why I stay in this job. That's really the only reason.
And if I can interrupt you, I want to leave 5 minutes to give people time to ask questions. Could you and Daniel shrink your answers to 1 minute each?
Yeah, I was gonna give it
Just
really.
speak really fast as a Colombian.
I was
Yes.
I was gonna answer the question completely differently, and it was already gonna be really short because I read this question very differently, more as if you were asking us what you think our superpowers as Latinos are. Is that okay? That's how I interpret it. So I think 2 things come to mind. One, we are very relationship based, and that's actually an asset. So that's an asset when teaching, that's an asset when interviewing, that's an asset when building collaborations. I would say that one. And then starting your lab, like, there's so many absurd situations that happen. And like, as Colombians, we're like super used to facing adversity. Everybody knows the 80s and 90s. When I was growing up, we're a rough time in Colombia. I feel like in all of Latin America, there's so much stuff that happens. And I feel like our instinctive response to laugh and just like move on, I feel like we don't tend to dwell on stuff. I feel like that's another superpower and something that I think my colleagues sometimes struggle with.
Well, that's what I was going to say, actually. But also, whenever I feel super proud, whenever I see some other person from Latin America, even the mouse, these Guatemalan mouse that did better than the rest, I'm like, yes! Every time some of these guys, in like yesterday's talks, they were all great. Like, I feel immensely proud. They have like these sassiness. They can be joyful. They, Americans, Latin Americans can be, very kind people. I feel proud every single time about that.
Thank you, and I'm sorry. I feel really bad about cutting people's answer.
My answer was going. I was going to give the same answer.
Thank you. Thank you for making me feel better, Anna. You're very kind.
You did cut me off, and I resent that.
Okay, we have 5 minutes for questions from people here. If there's anything that people want to ask people in the panel. Maybe we didn't need the 5 minutes, but I'm sure we do. Thank you so very much, Diana. And I have to repeat it for the people on YouTube. I wish we had like a microphone. Yeah. So you were saying that I'm amazing. No, just kidding. No. So Diana was thanking the speakers. I want to say that too, for being honest and open and sharing your experience. And I think that's very helpful for the community to hear, for us to bond with you, and also for everybody that's participating here. I hope this was a good event to bring people together, and hopefully something that we don't do just once. I mean, I would be very happy to reorganize it again next week. No, just kidding. No, just kidding. But thank you so much, Diana, for the comment. That was really kind. Questions or comments also?
Before Diana said that, I was going to say exactly that because it is, as a graduate student, as a postdoc now, you always think you go through all these struggles and they're yours and no one has gone through them and they're just yours. And hearing just people talk about them, I was very touched. I was almost about to cry because it's just relating to all of those experiences so important because you see faculty in a class, in a seminar, and you admire them, but just looking at how human they are and how they go through the same experiences as you, like you said, Anel, just like you, I can do that. I come from a minority, and so it is so important to like have those spaces for like just to see how inspiring everyone's like journey is. So thank you for opening this.
Thank you, Elena. And I'm just going to again repeat for the people in YouTube. So Elena was just thanking the speakers for sharing. And sometimes we feel that we're alone in the struggles that we have been, as an undergrad student, grad student, postdoc, faculty. But it's, I mean, I guess it's not good that we see other people struggling, but it's also it's also good to know that you're not alone in this. That people have overcome the challenges.
I also think it's important. So, for example, Laura, I admire her. She's like further ahead than me, right? And I always think it is so helpful to hear. She talked a lot about imposter syndrome. It's like good and important to know that it doesn't go away, because I feel like you just think, oh, at some point I'll reach a stage where like this won't matter to me anymore. Like I'll have it figured out. And the more people you talk to, you realize
I'll
that
let
nobody,
you know if it ever does.
yeah, like nobody does. So that to me it was also helpful.
Other questions or comments? Melina, I see you want to ask.
She's been,
yes,
yeah.
I know. Thank you so much for sharing, Melina. I think I'm gonna be a disgrace if I try to repeat all that. But Melina was just sharing what her experience of being in the middle and how she connected with Laura because of that. And I think I'm just gonna leave it at that if that's okay with you, because if I try to repeat everything, I'm just gonna say it wrong. Okay, we are time now. So thank you again all for coming. And if you have questions, if you want to reach out to people, if you want people's emails, I guess, if someone reach out to me and they want to get your email, maybe I can share it back. And thank you. Muchas gracias. Un placer.